Firstly, the brain sends an electric impulse to the muscles which are required to start walking, and this causes them to contract. This creates a force which pulls the tendons of a joint to cause the movement. There are 5 phases of walking. The first phase is the heel strike, where the heel hits the floor and does not move until the whole foot is planted. At this phase, there is a 30-degree flexion at the hip and full extension of the knee due to the contraction of the quadriceps. The flexion caused at the hip is due to the contraction of the gluteus muscles and the rectus femoris. The second stage is called the flat foot stage. The hip moves into extension as the body absorbs the impact caused by the contraction of the muscles, including the gluteus maximus. The tibiofemoral, which is a hinge joint, flexes, and the talocral joint plantar flexes to 15 to 20 degrees. The mid-stance phase occurs when the hip moves from 10 degrees of flexion to extension by a contraction of the gluteus medius muscle. The knee reaches maximal flexion and starts to extend. The ankle becomes supinated and dorsiflexed at 5 degrees.
Diffusion is the exchange of CO2 and O2 between the lungs and the capillaries that line the lungs. Carbon dioxide is released from the muscles and is transported in the blood to the lungs, where it moves via diffusion into the alveoli to be exhaled out of the body. At the same time, oxygen is inhaled into the body. This occurs through
The heel-off phase is when the heel is lifted from the floor. The coxal joint is at hyperextension at 10 to 13 degrees, which then goes into flexion. The knee becomes flexed at 0 to 5 degrees, and the ankle supinates and plantar flexes. At the early swing stage, the hip extends to 10 degrees and then flexes due to contraction of the iliopus muscle with lateral rotation. The stance phase is 60% and the swing phase is 40%. The factors affecting gait include stride length, footwear, surface, height and weight. The stride length is the distance between initial contact points of the same foot. Stride length is calculated by the distance times by 2 and divided by the steps taken. The cadence is the walking rate and is calculated as steps per minute. It is calculated by the number of steps times by 60 and divided by time.
oxygen is inhaled into the body. This occurs through oxygen entering the body via the nasal cavity and the mouth, before moving through the pharynx and trachea before entering the lungs through the bronchi into the bronchioles, eventually entering the alveoli. Oxygen then leaves the alveoli via diffusion, entering the capillaries before being transported to the muscles and organs. Gaseous exchange occurs across a diffusion gradient of high to low, meaning gas moves from an area of high concentration to low. Rate of diffusion is aided by several factors, which are the permeable membrane of both the capillaries and alveoli sacs, the thickness of both cell walls, which is only 1 cell thick, and the moisture of the membrane walls. Oxygen, when it diffuses into the capillaries, then combines with a protein called hemoglobin to form oxyhemoglobin, which is attached to red blood cells. This is how oxygen is transported to the working muscles in order for the muscles to work. During exercise, the oxygen consumption of the working muscles increases, which is why breathing rate in turn increases itself. Heart rate also increases during exercise. This is also to accommodate the working muscles. In order to carry more oxygen, more hemoglobin is needed. Therefore, blood needs to be pumped out at a faster rate. This is done through contraction of the chambers of the heart known as the atria and ventricles, which are controlled by electrical impulses. Impulses are sent from the muscles to the brain, which then sends impulses via the nervous system to the heart, which speeds up heart rate. The right atria contracts, pushing blood through the tricuspid valve into the right ventricle, where it is then pumped into the pulmonary artery and to the lungs. Once the blood arrives at the lungs and diffusion occurs, blood then arrives back at the heart via the pulmonary vein into the left atrium. Atrium then contracts, pushing blood through the mitral valve into the left ventricle before being pumped out of the heart via the aorta to the rest of the body and working muscles.
As stated before, cadence is the number of steps we take per minute, while our stride length is the distance covered between each step. Stride length varies from person to person and is highly dependent on height. We compared two participants of different heights and measured the difference in their stride lengths. We found that an increased stride length causes greater fatigue and physical exertion upon muscles in both tall and short people. However, in shorter people, the fatigue is greater due to the need to get longer stride length to match that of someone taller. This means that the fatigue in the quadriceps and calf muscles, along with the gluteus maximus, is increased whenever stride length is also increased. However, for our taller participants, the increase in stride length causes a lot less physical exertion as well as a lower increased heart rate when we measure the difference between the two participants. We measured the heart rate of both our participants in order to test how the heart rate is affected during different stride lengths, as simulated by a walk, a jog, and a run at 1 degree incline in order to simulate normal walking conditions. We found that an increased height meant the larger stride length was more natural. It was easier for the participant to run for a sustained time as their heart rate was lower than that of the shorter participant.
When we walk along the sagittal plane. Walking constitutes a series of repetitive cycles comprised of 2 steps. At the coxal joint, during the swing phase, the joint is in flexion, while during the touchdown phase, the coxal joint is in extension. This is the same for during the heel rise prior to toe-off. At the tibiofemoral joint, it is at flexion during the touchdown phase. The joint is then in extension at heel rise and flexion during the toe-off to swing phase. While during touchdown, the tibiofemoral joint changes from flexion to extension. The graphs from KA2D show the time of the greatest angle during walking of the joints we examined. This allows us to determine the angles of specific joints during the movement pattern. While walking, different forces have different effects on the body. Forces such as air resistance, gravity, and friction determine the speed at which we walk, in which we can determine the distance travelled. These are the joints that we're going to be measuring the angles and distance for. This is
the glenohumeral joint,
this is
humeral radio-ulnar joint,
this one is
the radiocarpal joint,
this one,
the coxal joint,
this one is,
the tibiofemoral joint,
and this one is. We use the 1 metre rule in order to scale to find the length between joints and angles. Different forces have different effects on the body. Forces such as air resistance, gravity, and friction determine the speed at which we walk, which is then determined by the distance covered divided by the speed in seconds it takes the participant to cross the distance. In order to compare the difference in stride length and cadence for our 2 participants, we need to take anthropometric measurements, such as height. The angles then were able to be calculated between the different joints. It allows us to compare between the 2 participants to different stride lengths, cadence, and speed. limitations to the way we have conducted our assessment. Firstly, while filming, the background has to be bright enough to be able to see the participants clearly, while the shutter speed and aperture have to be set properly to avoid overexposure. We found that during our digitisation, the frame rate was a little slow. At some points of digitisation, we found it hard to distinguish points of the body. We required to fully and accurately digitise the angles and images. Other limitations during filming were the clothing of the participants and the markers we used. Baggy clothing can move during filming, which means that the markers would not always be at the joint and would cause inaccurate digitisation. The markers themselves cannot be the same colour or a similar colour to the clothes, as they cannot be distinguished away from the clothing, therefore have to be bright and able to be viewed from a distance by the camera. In terms of the limitations for the testing themselves, the most inaccurate was the measurements of the heart rate and timing. We used heart rate monitors to measure the heart rate during the different walking stances, e.g. difference in stride length and normal walking. We also used the heart rate monitor to test running to show different heart rates between different people. We found the heart rate monitors go significantly higher reading of heart rate rather than when taken by palpation, showing that they can be inaccurate and lead to false readings. As far as timing, using a stopwatch is based on a person's reactions and can be inaccurate, whereas using timing gates would provide a much more accurate time. The more the timing gates are, the better the measurements we can get, such as acceleration, deceleration, and the average speed of the participant.
The first stage in which we prepared for the use of the camera was by placing down tape on the floor, perpendicular to the camera, which was placed at a 90-degree angle. To show that this is correct, the Pythagoras triangle can be used. We then place the camera on a tripod so that it is positioned correctly. When setting up the camera, a plumb line is ensured so that it is directly above the perpendicular line. The height of the camera should match the midpoint of activity or same height as the individual's hip. Once this has been carried out, we must ensure that the background is clear, that it is non-reflected and does not contrast with the performer. Also, we must ensure that the field of view is uncluttered. Once the equipment is all set up, we must ensure that the individual is in the midpoint of the field of view by zooming in to the correct length. Finally, we label the individual's joints with coloured tape in the shape of a square. Focus and field of view. The focus of the camera is manually set and not automatically focused. To manually focus the camera, we had to place the individual in the field of view and zoom in or out to correctly focus the image. By using the focus tool on the camera, we are able to correctly focus the individual. Once focused, we adjust the zoom of the picture to the appropriate size. This therefore avoids excess space and bad focus. Scaling. When scaling the camera, it is important that it is recording where the participant will be performing. There are two types of scaling. There are horizontal and vertical scaling. When using these types of scaling, they must have obvious endpoints. Aperture is the opening in the lens which restricts how much light is able to reach the image sensor. There are two different types of aperture. There are small and large. Small aperture is where the light is underexposed, whereas large aperture is where the light is overexposed. Additional lighting may be needed even if the correct shutter speed has been set, for example a floodlight. The aperture is auto-adjusted by the camera itself. In photography, shutter speed or exposure time is the length of time a camera's shutter is open when taking a photograph. Both shutter speed and aperture control expose the image. Fast shutter speed is better for high speed movement and is more clear.